Hello and welcome, dear friends, once again to our discussions on tools for molecular cloning. In my last lecture, I had given you a fair idea about the use of plasmids as vectors for molecular cloning. Today, we will see the use of phages, bacteriophages, with special reference to Lambda phage as a vector for recombinant DNA technology or the molecular cloning. The need for vector basically arises as a prime requisite for this process because vector is an agent which carries a DNA fragment into the host cell, and that host cell which is capable of replication. If you see the process which I have been repeating again and again for your understanding is restriction endonucleases, which is used to cut the DNA. The DNA or the sequence of interest, the gene of interest, the same recombinant DNA molecules symbolized by the scissors here, as they are already known as molecular scissors. So the same restriction endonuclease is used to cut the vector also. So the restriction enzyme cut of the DNA and the gene of interest makes the both end compatible so that this DNA of interest can be introduced into the vector, and this vector containing the DNA of interest is multiplied into multiple copies. We say it is cloned. So if the vector which is the agent for carrying the gene of interest is to be used, with time, with advancements in genetics, just like I would like to give here example of your smartphone or the mobile phone as compared to the mobile phones of back 10 years, a decade long back. If you see, the memory was very small. With increased smartness in the phone, with increased smartness in the technology, you need more and more RAM. That is, first it started with 1 GB RAM, 2 GB, 3 GB. Now we have 8 GB RAM. Once upon a time, it was even impossible to imagine a complete desktop function station without with this much GB of RAM. Today we are having a memory of size that much in a small phone. Same is the case with vectors. With increased technology, with increased efficacy of the process, with newer understanding of the genome and the molecules as such, the need to insert larger and larger DNA fragments is becoming the need for genetic engineers. So vector should be an agent which should be able to carry a DNA fragment of the interest as long or as large as possible, because it will be a polycistronic molecule. It will be a multiple protein encoding molecule. Okay, and it should be capable of replication. So if it is used, if such type of DNA and the vector combination, and the vector is used only for reproducing the DNA, we call it as a cloning vector. Like this, only the purpose of this vector is used to replicate the gene of interest, it is a cloning vector. But if it is used to express the gene itself within the vector, within the host, it is used to express, then it is called as expression vectors. There are various types of vectors: plasmid vectors, bacteriophage vectors, cosmids, bacterial artificial chromosomes, BACs, yeast artificial chromosomes, YACs, HACs, human artificial chromosomes, PACs, P1 phage derived artificial chromosomes, mini chromosomes, so on and so forth. As I said, with increase in time, with increase in the knowledge, information, database and understanding, newer vistas, newer windows have been opened in this technology, and with every passing day, who knows, my lecture this will be absolute within a year or two. Maybe this will be history, what I am speaking today, within a decade or so. That is the advancement which this science in the field of molecular biology is seeing nowadays. So the urge to accommodate larger and larger DNA fragments inside the vector is changing the basic definition of vector. So in this series we have seen the grand old daddy of vectors, which we call it as PBR 322 plasmid. Similarly, Lambda phage, this is one magnificent phage for in its own world, in its own kingdom, which has a miraculous type of lifestyle, a dual lifestyle, and so sort of it can even remain in a lysogenic state for as long as it can see the favorable conditions and then break open the host and come out after lysing the cell. So two types of lifestyle, a large DNA fragment, and some of the features why it is considered as a very good candidate for using it as a vector, we will see in this lecture. So bacteriophages, specifically lambda phages as vectors. Bacteriophage is any of that virus which has bacteria as a host, which can infect bacteria. Here Escherichia coli is the host of this lambda phage. Commonly based upon the lambda phage, it is a virus particle which consists of a head. As you can see, the whole functioning here, it consists of a head in which the DNA is packed. It is a complete nucleoproteinaceous structure. It consists of a head, a double-stranded linear genetic material, DNA, double-stranded DNA as its genetic material, then a tail sheath, a long cylindrical tail sheath, tail fibers, and this is for attaching on the host, and it has a very beautiful mechanism of attachment, then just sits on the host, injects its DNA, and the ghost body is thrown out. So this is the structure of a typical tailed bacteriophage. I will not go into much details as you can see these things in virology. The point of discussion here is using this parasite, obligate parasite of Escherichia coli, as a molecular tool for transferring our gene of interest, not only transferring but using it as an expression vector, using it as a candidate. Now we redesign it, we redesign its genetic material. How that is done, interestingly, we will see in this lecture. So if you see the lifestyle of lambda bacteriophage or the lambda phage, it can enter into dual mode. Of course, the ultimate fate is the lytic cycle. It always strives for entering into a lytic mode and making multiple copies of itself. If it is in its utmost favorable conditions of infection, means whenever there is a host and a parasite interaction, the question is who is gaining the upper hand. If host is gaining the upper hand, the host immune system will not allow the phage to multiply. In that case, very slyly, very cautiously, the lysogenic mode is activated by the phage. It does not allow the maturation proteins like head, tail, fibers to grow. It just integrates itself into the bacterial DNA. Again, it has a dual mode there also. It can integrate or remain unintegrated, entering into a abortive type of transduction. Those are the very interesting aspects of bacterial viral life cycle of a bacteriophage, which you can study apart from this discussion. So here the point is, it can enter into a lysogenic cycle where it can integrate its own DNA into the bacterial DNA and survive as if nothing has happened till the favorable condition is reached, where then suddenly, as you can see this dotted arrow, one day it will again enter into the lytic cycle. Now this is the path which we are going to explore, that is the integration of the phage DNA into the bacterial DNA. That is what we are going to exploit as the molecular tool. So that was just for your information. Now these bacteriophages, when these are used as vectors in the recombinant DNA technology or molecular cloning, one interesting aspect is, it was discovered that most of the internal genes, here the violet part as you can see, this lambda phage, when it infects a bacterial host, a E. coli host, it injects its DNA, this is the complete lambda DNA injected. There is a central gene cluster. What part is that? You will see in the further slides. That can be removed. Now of course I am talking about in vitro process. We can take these phages, wild type phages, and remove the middle region. This is called as the stuffer region, and this can be replaced by the pink colored DNA, that is the gene of interest. The insertion of gene of interest, and then this DNA can be packed in vitro. This is called as in vitro packing. It can be packed inside the phage heads and recombinant viral particles. These are the recombinant viral particles which are then now able to infect a bacterial host, and these are carrying the cloned DNA fragments, and then they will replicate. How to see whether these bacteria are dividing or not? You have taken out the unessential genes and introduced your gene of interest into the lambda DNA, and then it is allowed to multiply in a host. Now this is checked by, as you can see, this is a real plate which contains a lawn of Escherichia coli bacteria, and you can see plaques here. These are plaques. Plaques are the characteristic features of a bacteria growing in a lysogenic condition or a lytic cycle. When there is a lytic cycle, you can see clear plaques here, complete clear plaques. And if there is a sort of lysogeny or incomplete lytic cycle, you see a cloudy or a translucent plaque. This is a cloudy plaque, not so clear. So these not so clear plaques are indicative of your gene of interest getting totally integrated and the cloned vehicle getting expressed. So this is how the bacteriophages they are used. Now cloning vectors which are based on lambda phages or the bacteriophage lambda, there are two problems associated with it. Means, what is our aim? Our aim is to use lambda phage as a vector. We are trying to replace its original length of a DNA with our gene of interest. Now you have to take care that its replication proteins, its multiplication proteins, head and tail protein should remain intact. Fortunately for us, we have a length of a DNA, we have certain region of a viral DNA which are really non-essential. Means if you remove that, its normal life cycle maturation will not be hampered. Some of the genes are there that can be removed and you have to insert it. But before considering this, the cloning vector, if you are thinking of inserting the gene of interest directly into the lambda phage, it is not possible. Two problems have to be solved before lambda based cloning vectors can be developed. The first problem is that you can see the size limitation. This is the first problem. Normal lambda genome is 49 kilobases in length. 49,000 base pairs it has in its DNA, double stranded DNA, which is packed inside a phage head, and it is called as a head full of packing. Means if you have a 1 liter glass jar or a beaker, you cannot even think of pouring 1 liter and 10 ml inside it. Why? Because its capacity is 1 liter and the complete up to the brim filling is 1 liter. Even if you add 1 ml, it will spill. Just like that, it's called as a head full of packing. Head full of packing allows 49 kilobases of a DNA to be packed inside a phage head. Now usually, even if you think of considering a gene of interest as small as 3 kilobases, that will increase the gene size of this lambda to 52 kilobases, and that will not be accommodated. The DNA molecule will be increased in size. It can be increased only up to 5%, and if this is 3 kilobases, as considering 49 kilobases, the 5% is 3. The total size of the molecule will become more than 52. So it cannot be packed into the head structure and infective phages cannot be formed. Means they will not multiply. So this particular first point, this severely limits the size of the DNA fragment which can be inserted into a unmodified vector or a non-modified vector. What is that modification? As I told you earlier, you have to remove certain regions in the middle, which is called as stuffer region, which I will discuss in the next slides, coming slides. So basically, when you start to use a lambda phage as a vector, you encounter two problems. What is this? This is the first problem, that is the size limitation. The second problem is that there are multiple restriction sites, that is the restriction endonucleolytic cleavage sites. For example, you take EcoR1 enzyme and this is the length of the lambda phage. If you are using restriction endonuclease or the restriction endonucleolytic cleavage on the phage DNA using EcoR1, there are at least 6 points by which you can generate 6 fragments of varying lengths, which will automatically religate because of the same restriction endonuclease making cuts on the DNA, each can religate, means one can ligate and tail end ligation it is called as. There is end to end ligation. As all the ends are compatible with each other, as it is being cut by the same restriction endonuclease, they can ligate. 2 can ligate with 6, 5 can ligate with 6. So there will be a random religation which will give rise to a complex mixture of molecules. So you never know what is the part where you are going to insert the gene of interest. So the lambda genome second problem is multiple restriction site problem. The lambda genome is so large that it has more than one recognition sequence for virtually every restriction endonuclease which can be used here. The example given, even though if it is EcoR1, you can use any restriction endonuclease enzyme and it has multiple recognition sites, and this restriction cannot be used to cleave the normal lambda molecule in a way which will allow the insertion of new DNA, because this cleavage will not allow insertion of new DNA, because the molecules will be cut into multiple small fragments and they will be very unlikely to form a viable lambda genome on religation. Means the head genes, tail genes, the maturation proteins, you never know where they will religate. And a viable means which will be acted on by a promoter, and then transcription and translation has to take place of this DNA which will form rise to heads and tails. That viable religation is not confirmed or is not guaranteed here in this case. This is the second problem, but these are addressed. Now these two problems are the problems which we are considering at the beginning of using this as a vector for molecular cloning. Now how these problems are solved, let us see. If you see the characteristic features of bacteriophage vectors, some of the features we will see. These are bacteriophages. Lambda phage is a bacteriophage. Its genome is a circular genome and it has a cos site. It's called as cos because it is a cohesive end. The cohesive end point, it can be cut in the middle and becomes a long length of a DNA. As you can see, this becomes a long length of a DNA. It is cut to cos sites, and both ends they have 5' end and 5' end cos sites. These are called as cos sites. It becomes a linear DNA. This is the beauty of the lambda phage DNA, and within this there are certain all the genes, head genes, tail genes, recombination genes, integration genes, head, tail, recombination, DNA replication, lysis, so on and so forth. So this is the genomic map of lambda phage. These bacteriophages or phages, they are known viruses which infect the bacteria, you know very well. So like viruses, phages, they are very simple in structure, consisting only of double-stranded DNA, that is a lambda phage. Lambda phage is an example. Bacteriophage M13 are another very good competent vectors which are used. Unfortunately, we don't have that in your curriculum, so I am not discussing it here to avoid the length of this video. So you can study that, as those who are interested in competitive examinations, you can study the bacteriophages M13 as vectors. They are also equally competent vectors as example of bacteriophage vectors. We will be discussing lambda phage vectors which are widely used for cloning because they can be used to carry large fragments of DNA as compared to plasmids. Larger segments of gene of interests can be introduced as compared to plasmid vectors. Lambda is a typical example of head and tail phage, and the lambda molecule, the genome as we just saw, is 49 kilobases in length. There are two types of lambda vectors, that is lambda insertion vectors and lambda replacement vectors. What are these? I will explain. The advantages of using lambda vectors as a tool for carrying your gene of interest is that the storage of phage particles is comparatively very easy as compared to phage plasmid based vectors. The storage of phage particles means you can store it on nutrient agar plates in the form of plaques. You can store it in liquid cultures using Escherichia coli or the bacterium as a host, so on and so forth. It's easy to maintain these phages by supplying the host. The shelf life of these phage particles is infinite. Means as long as they get the desirable conditions like temperature, host, etc., they will remain in a dormant stage and they can be used to store for long durations of time under proper virological microbiological conditions. The transfection of E. coli is much easier with phage particles, means from one host to another host within the strain, or just transfer of this DNA from one bacteria to another bacteria, like transduction. Now we are not calling it as transduction, we are calling it a transfection because we have a designed genome in this. So transfection is the infection of this cloned viral particle from one host to another host is very easy. You just have to, anyway, viral infection is very easy. It is almost automatic when there is a healthy host. Understand this point. If a host is healthy in the sense with respect to, we can modify that, you remove its immune system, host restriction and modification system which is there in E. coli, if it is those genes are removed, then you can carry out a very easy infection. We call it as a transfection of these phages from one host to another host. That is advantage of using lambda phages. Now the only disadvantage of using this lambda vector is it is difficult to isolate. Just like the colonies which we can transfer from one plate to another plate with just a nichrome wire loop, that easy process is not available for isolating a virus from the host. You need hosts, you need very specific hosts and purity for this. But still, today I don't consider this also as a big hurdle, because in the advancement in technology and with skilled human resource development, we have overcome this barrier also. There are certain characteristic features of lambda phage. As I have said, it is a head and tail phage with a 49 kilobase of DNA. Esther Lidberg, he found E. coli K12 strain to be lysogenic for lambda phage, and this marked the discovery of lambda phage as a phenomenon for using it as a vector, lysogenic vector. Okay, lambda phage as well as he also saw that the lambda phage undergoes lysogeny, and this became the landmark or the point where it was decided that it can become a good vector for recombinant DNA technology. Some of the character features of this phage are: it is a temperate phage. Temperate phages are those phages which can maintain themselves in lysogenic state without immediately lysing the host. This is a very wonderful feature, a very handy feature in the recombinant DNA technology. Means if a phage is infecting and instantly the host is dying, it is undergoing lysis, then that process is of no use. The temperate phages are those phages which have the capacity to undergo lysogeny. Now this is a lambdoid phage. What is a lambdoid phage? Which resembles the Greek letter lambda. If you see the Greek letter lambda structurally in terms of design, just like a lambda is there on a tripod, just like this phage is with the head and tail, and it resembles the Greek letter lambda. Therefore it is a lambdoid phage which has a host E. coli. Therefore it is a coliphage. It's called as a coliphage. It belongs to Cephophyridae family, nearly 50 nanometer in diameter. And you can see in the screen here, just pause and take into details about its physical dimensions, and that is just for your information. Of course has very less significance when it comes to being used as a molecular tool. Anyway, it is going to infect a E. coli, and the burst size is around 300 to 1000 phages per bacterial cell. It has an icosahedral head and a flexible tubular protein tail. As you can see in the figure, it is flexible, and a connector. It serves as a site for attachment for head to tail, and these fibers, these are used to attach on the cell wall surface of E. coli. Now in the DNA, if you see its DNA, it possesses a cos site. This cos site is called as the cohesive end site. In my next lecture, I will be discussing about a vector called as cosmid, and this cosmid utilizes this cos site. It's very interesting, but that will be the next part of our lecture. Here, it possesses cos sites at the end. If it is cut in the middle of the cos site here, it becomes a linear DNA, and at the two ends of this linear DNA, both will have cos sites. Okay. And its molecular weight is around 31 into 10 to the power 6 Dalton. The protein, as it is a nucleoproteinaceous structure, the protein is of that size of that dimension. And as I said, due to the presence of cos sites, that is the cohesive end sites, DNA it adopts a circular structure when it is to inject into the host. And while using it as a molecular clone or recombinant DNA technology, you can make it linear also. These are some of the characteristic features of Lambda Phage. Now I have already shown you the lytic and lysogenic mode of the bacteria. One interesting point which has to be understood of this lambda genome is how the DNA phage is inserted into the bacterial genome. Now that particular character is used to introduce your stuffer region or replacement, and the gene of interest with the lambda is used to insert into a E. coli chromosome. So if you see in E. coli, there are two genes, galactose and biotin, which are in tandem to each other. Means it is linear, one after the another. Galactose and biotin gene are there, and in between that there is a small region called as attenuator region. The same attenuator region is present in lambda phage. This is a very interesting thing in the life cycle of E. coli and lambda phage. You can just question, why is E. coli genome containing a attenuator site which will allow the parasitic integration? Think about it. A very fascinating question. Its answer is as fascinating as it is how the host and the parasite, they evolve during evolution. Evolution is a long process. They have been fighting, maybe lambda and E. coli, they have been fighting for long, and they are fighting for adjustment. So they have come to a point of compromise where the E. coli, it allows the integration of lambda phage so that for some generations it can multiply. That is how we have developed our immune system with the viruses. Take a break here, point in time, take a break for the discussion about lambda phage. Let's go into our immune system. If you think we have thousands of different types of viruses in our body, how we are then immune to it? It is a compromise between the viruses and our immune system. They have adapted. Same may have to happen with the coronavirus also. Today we are seeing huge population being destroyed by the infection of novel COVID virus, coronavirus. And when will this stop? It will stop when our body will start adopting to the viral genome, and we will come to a point of compromise where we will allow it to reside in our body, and occasionally it will go on bringing some fever and some chills, and it will go. Similarly, come back to our point of discussion. The phage DNA gets integrated into the bacterial host DNA at a point called as attenuator, and that attenuator region is also present in the lambda phage, which it has developed. See that red point? That is attenuator point in phage. Attenuator P is attenuator phage, attenuator B is attenuator bacteria, and it has reversal integration point. Means if you say here 1 and 2, and if you call it as 3 and 4 at the point, here 1 and 2 on B and 3 and 4 on P, this 4 will attach with 1 and this 3 will attach with 2, and there will be a reciprocal type of integration inside the bacterial genome. Phage genome will reciprocally integrate, and it will remain there as if it is a part of the bacterial genome. This is how the DNA of the phage is inserted into the bacterial genome between galactose and biotin gene. And that is the point. The cos site is there responsive. There is a attenuator site, near the cos site it makes a cut and it gets reciprocally integrated. This is the beauty of lambda phage genome. And once it integrates, it can transcribe and translate to the left as well as it can transcribe and translate to the right. It is a very master DNA molecule which has evolved with times in multiple genetic functions. Okay, all in all, as you can see, there is integration, there is excision during insertion into the bacterial genome. There is integration, there is excision, there is a reciprocal type of integration, and all are carried out with the genes which are accommodated in the phage DNA. All these things, all these features makes it an excellent tool to be used as a vector. Okay, so some of the features as a cloning vehicle, we just saw. What we saw is the features of the wild type Phage genome. Now we will see some features as a cloning vehicle. Why it can be used as a cloning vehicle? It has a large genome size. It can be used as a cloning vehicle because it has a large genome size and low insert capacity. Means the insertion of the gene of interest, if you are trying to insert it in a wild type genome, it has very less size, but if you remove a certain region, the insert can be made larger. Head can accommodate around 49 kilobase pairs of DNA. This is one of the feature of cloning vector. Means now you can remove some genes and you can insert, and around 49 to 50 kilobases of final DNA. Of course, there is a limit to the size of gene of interest, because you have to retain the head genes, tail genes, fiber genes and replication origin genes for the multiplication of the phages, so that your DNA also replicates. And that is allowed in the bacteriophage when we modify these things as a genetic engineering. It allows the side reduction. As you go on cutting the longer lengths of Phage genome, larger lengths of gene of interest can be inserted. So it allows size reduction of the wild type genome to increase the cloning capacity. Only 50% of the genes of the wild type Phage, these are essential for replication and lysis of the host cell. Try to understand, give a thought. 50% of genes are only essential for it to multiply, etc. What are the other 50%? This is for maybe evolution. So we can remove that and you can replace this 50% with your gene of interest. Okay, that is one of the characteristic features of bacteriophage lambda, why it can be used as a cloning vehicle. Then it allows the alterations in the number of cleavage sites for restriction enzymes. There are multiple restriction cleavage sites in it, which makes it an excellent tool for molecular cloning. Then natural selection of the modified lambda phage which lack these certain genes can be made. Means you can naturally select its original feature, like the plaque formation on the plate, that can be used. Okay, then recombinational insertion of region from other lamboid phage can also be done. Means you can take lamboid genes from other strains and you can club it. Why? You can even take the lactose gene and stuff it here, so that you can use a lactose to regulate the lawn of bacteria. Multiple options are there. In vitro, the genetic recombination leading to the exchange of immunity regions, means, as I said, host restriction and modification of the DNA, and which encounters that can be removed by in vitro mutagenesis and in vitro manipulation of the genes of this phage. So all in all, it allows manipulation, it allows mutation, it allows modification of 50% of the genes, which makes it an excellent cloning vehicle. Now if you see the gene organization of lambda phage, we will go to the lambda phage genome, and I want to emphasize here which part of the genome can be removed. So if you see, there are various genes which are resulting into the process of recombination, that is integration, which I just showed. Recombination is for prophage formation, reciprocal integration. Then this is the control region, the CRO genes. These are the operator region and the promoter region, which is also used for replication. The late control, lysis genes, virus head and tail formation genes. So this linear gene is coming from the circular genome, which is joined at the cos sides. This circular genome, if you just cut at the cos side, come to the left side here, you will get a linear DNA which has cos site at the two end, and in between which essential genes required for maturation, replication, recombination, etc. are present, which is shown here. Now here in the yellow region, the same I have simplified here. Two ends there are cos sites. Now this is one DNA. The same DNA can be represented like this on the left. The same DNA can be represented here in the colorful strip, which you can see. The same DNA is simplified here, showing all other genes, and a central region which is called as a stuffer region. This stuffer region contains two restriction sites which can be cut, and this can be removed and replaced with our gene of interest. So if you see the genome of genetic organization of lambda phage, it has a left hand region. it has a central region and it has a right hand region. The left hand region, if you see, it encodes for head, tail and proteins. Of course, the left mirror, as it is a mirror image on the screen, this is the left region, this is the right region. The left hand region has genes which has coding for head and tail proteins. These are called as genes NU1 and a series through J. Central region, this is the central region which is J through GAM. Here you can see the GAM, which encodes for integration, recombination, and there is a attenuator site here, ATT site, which is an attachment site. Okay, so this is the original wild type gene organization. I will now tell you which is the stuffer region which can be removed. But before that, see, there is a right hand region which has NQ genes. This is the N gene, this is the Q gene. Between the N and the Q gene, which are for lysogeny. Genes through N and Q, these are lysogeny and lysis. There is an operator gene and there is a promoter gene. There is a DNA replication gene. Then S, R and RZ are the host cell lysis genes. So the point here is this, all these are essential for the Lambda phage to infect, inject, recombine its DNA or provoke the host cell to multiply it in number and then undergo a multiple copy formation of the phages, bursting it into lytic cycle. This is the normal mode. Now see, there are certain segments of this genome which you just saw which can be deleted without losing the viability of the phage. I hope you are getting the point. You can use this genome, the whole DNA sequence of the lambda, cut it in size up to certain region. As you can see here in the figure, there are certain non-essential region from around 15-16 region of the on the genetic map, 15 to 35 position. Now these are about the kilobases, around 15 kilobases to 35 kilobases on the genomic map. This part is called as the non-essential region. Now you see this word, it's a very confusing word. Non-essential, it is essential for the virus, but when we are using it as a tool for recombinant DNA technology, we can render it non-essential and insert our exogenous DNA or the gene of interest DNA or the clone DNA, which we are going to use it for the recombinant DNA technology. So there is a large segment in the central region of the lambda DNA which can be removed without affecting the ability of the phage to infect the E. coli cells or the replication also, formation of head tail fibers also. Removal of all parts of this non-essential region between the positions 15 to 35 around, this is approximate, you can get different numbers on different literature, but broadly speaking 15 to 35, resulting in a molecule of around 20-23 kilobases. This means that here you can see approximately 20 to 23 kilobases DNA can be cut off before the packaging is reached. In between the left long arm and the right short arm, there is exogenous DNA which can stuffer region, which I call it as, or the non-essential region. It can be removed. This non-essential region in fact contains most of the genes which are involved in integration and excision of the prophage. Think about it. The integration, and which I said, that is the original wild type mode which is there. Now as I said, this region can be used in recombinant DNA technology, means used by removing that and replacing it with the gene of interest. We are not needing this phage DNA to integrate in the host. We want it to multiply in a form of lytic cycle. We want it to multiply and replicate our cloned DNA. So it becomes a cloned viral particle. So this non-essential region which is needed for integration and excision, that is, I will go back one slide. See here, this genes, integration and recombination, this at region. Okay, how come this is not the central region? This is not the stuffer region. This becomes central when the two ends are joined. You see this circular DNA. Now this is the region. This is the region recombination, integration. This is removed. So if you see this DNA, this is the central region around. As it is a cyclic permuted DNA molecule, you cannot call it as central. It is one region of the DNA, this region, which is containing, come back to our slide, which is called as the non-essential region needed for integration and recombination. It is removed, and this deleted lambda genome now becomes non-lysogenic and can only become lytic, which is needed. So its lysogeny part is stopped. When I said this part is used, the lysogenic cycle nature is used. Used means that region is cut off and used for stuffing the gene of interest. I hope that is clear. We use the lysogenic gene in removing. This is the exogenous DNA which is used for lysogeny, integration, excision, and it is not needed for us in the recombinant DNA technology. So we just use it for stuffing our gene of interest. And that is how these segments of the lambda genome can be deleted without losing the viability of lytic cycle, and that is how you create a clone, or that is how the use of bacteriophage lambda in recombinant DNA technology is carried out. What are the steps in cloning the lambda vectors? You can see this stuffer region, it is restriction digested by the same enzyme. This stuffer region is released. The same length or plus or minus 1 to 2 kb is allowed, as we have seen in the earlier discussion. Up to 53 kilobases can be accommodated. So here a short length is removed and insert. This green region is our gene of interest, which is inserted, and you have a recombinant DNA which will be now packed inside the in vitro packing of the phage, a head full of packing, that is called as. So the first step is restriction in digestion of the lambda vector as well as the genomic DNA. This genomic DNA, which is green, and the lambda vector, which is the yellow stuffer region, is acted upon by the same restriction endonucleases. Then it is ligated into the DNA of the lambda genome. So ligation of the DNA fragment into the lambda vector, which forms a recombinant. Now this recombinant is introduced into the host by a process called as transfection. Competent hosts, they are transfected, means there need to be a very specific type of phages, and its host strains may vary. They are used. These are called as competent host, and transfection is the process. So E. coli is the host, or in vitro packaging of the recombinant DNA is done by addition of this packaging extract, and a natural infection of the host is done. Then selection and screening is done by plating techniques. You can go for the plaque, clear and turbid plaques, or there are various types of color dyes which are also available today in the market of recombinant DNA technology tool selection. Okay, so this following considerations should be made which will influence the selection of this vector. First, restriction enzyme to be employed. There should be sufficient restriction endonucleolytic cleavage sites, one thing, and it should be compatible with the gene of interest. Size of the foreign DNA to be inserted should be determined. All these are carried out by the gel electrophoretic techniques. Type of screening to be employed should be known, and expression of the clone gene in the E. coli. This is a very important factor which influences the selection of your vector. Okay, so what are the factors which influences the selection of lambda phage as a vector? Whether it should be selected or not depends upon the restriction enzymes, depends upon the size of the gene of interest, depends upon the screening system. Means after the whole thing is done, there should be some method to identify whether this has been properly inserted or not, and expression of the clone genes. These are some of the issues. Now there are various classes of lambda vectors. Two classes predominantly: insertional vectors and replacement vectors, or substitutional vectors. I'll give you some examples. This is very simple. Now whatever the type of gene of interest you are using, either of these regions, that is recombination, integration and excision, of which is helpful for lysogeny, is removed. So when you insert a small DNA, that is vectors which contains at least one restriction enzyme and allow the insertion of a foreign DNA are called as insertional vectors. You can see this is the wild type lambda genome. So lambda phage, when they are used as insertional vectors, you can see at the C1 site, which is one of the insertion, echo R1 is acted. Now there is a cut and various inserts from 0 to 10 kb only. Insertional vectors have their size limitations. Size of the insertional vectors cannot be below 37 base pairs, kilo base pairs, and that of a foreign DNA, means the original size of the insertional lambda vector cannot be below 37. So means it allows not more than 10 types, 37 of the lambda phage and 10 of the gene of interest at the maximum. Okay, so size of insertional vectors cannot be below 37 kilobases, so that the foreign DNA should not increase after addition above 52 kilobases. So you can have inserts from around 1 kilobase to 10 kilobase only. Okay, such type of vectors which range, there are different examples like lambda GT10. GT10 has 10 kilobases, GT11 has 11 kilobases, so on and so forth. But these 18, 23 need not be the base pairs or the kilobases, they can be the series numbers also. So construction of a lambda insertion vector. Two types of vectors, as I say, insertional vectors and substitutional vectors. In insertion vector, the normal DNA is 49 kilobases. After cleavage and ligation, you convert this 49 kilobases, non-essential region is removed, and it can be in between 35 to 40 only, around 37 kilobases it becomes. And in this 35 to 45 kilobases, that is a short lambda genome, you can now insert around 1 to 10 kilobases of your gene of interest. Insertional vectors. Now second, before going to the second type, let us see some examples of phages as insertional vectors. These are lambda GT10. As you can see, lambda GT10 as a insertional vector, it carries up to 8 kilobases of a new DNA. It is inserted in a unique echo R1 site near the C1 gene, of the lambda genome. Insertional inactivation of this gene is done, which means that recombinants, they are distinguished on the basis of clear plaques rather than turbid plaques, because lytic cycle is very much clear when this is carried out. So lambda GT10, as I said, one of the factors influencing the use is how you are going to detect it. So it gives clear plaques. One of the features of this is it gives clear plaques. Okay, so this is one. Then there is lambda ZAP2. ZAP-Z is use of a bacteriophage LAC-Z gene. Very interesting. See this. Creation of a lambda phage where the bacterial LAC-Z gene is used, in which insertions about 10 kilobases DNA to any of the 6 restriction sites with a polylinker. I have explained what is a polylinker, which inactivates the LAC-Z gene, is carried by the vector. Means LAC-Z gene is used for insertion, and that is seen whether it remains active or not. Okay, so what happens if LAC-Z gene is inactivated? It will give white or blue plaques rather than the clear or turbid plaques on the plate where you are growing it on a lawn of host where E. coli is the host. So if in E. coli, in the host, if there is no infection by the virus, then that will give a different type of color, and but if that is host is going inside with the LAC-Z gene, it will give a different color. So here the recombinants give clear rather than blue or white plaques. Blue plaques, that means that no infection has taken place, and you are using this LAC-Z gene, okay, as one of the polylinker which gets inactivated when it is used for insertion. So these are the two examples of lambda phage as insertional vectors. The next is replacement or the substitution vectors. Substitution vectors, this is substituting the non-essential part of the vector with the gene of interest. As you can see, there was insertion, here there is substitution. A complete region is replaced. Rather than shortening the DNA, on the basis of size, and inserting considering the size, you just replace the DNA, cut the DNA and replace. So you get the original length of the phage, original genomic size in kilobases of the lambda phage is retained with the gene of interest. So that is lambda phage as substitution vectors. Substituting the non-essential part of the vector with the gene of interest is done. That replacement gene is called as the stuffer fragment. Lowest possible size of the replacement vector can be 37 kb as same. So there are various examples. This can be removed and around 10 to 20 kb of inserts can be done. So you go one step ahead. In the insertion vectors, you have 1 to 10 kb of gene of interest interests which can be inserted. Here it allows 10 to 20 kilobases of gene of interests which can be inserted. So these examples, they are Charan series. Just remember the names. Charan series, these are substitution vectors of lambda phage vectors. Lambda EMBL series, lambda GT series, lambda C series, lambda GEM11, GEM12, lambda dash, lambda fix. These are some of the names derived from coming from various laboratories which have evolved into a very good lambda phage vectors. So this is the basic process. As you can see, this is the original wild type DNA. See the red blinker here. The stuffer region is removed. It is restricted and ligated with your new DNA or gene of interest. Now it allows a larger length of gene of interest because you are replacing rather than substituting, rather than inserting, you are substituting the gene of interest. So this is called as a substitution vector. So example of lambda phage as a replacement vector, lambda EMBL4. This is one example which I would like to give. It has 3 restriction endonucleolytic cleavage sites. EcoR1 coming from Escherichia coli, BAM coming from Bacillus amyloliquefaciens, and SAL coming from Salmonella restriction endonuclease. These are EMBL series. It can carry up to 20 kilobases of inserts of DNA by replacing a segment which can be cleaved by these 3 restriction enzymes. So that is called as the RBS or the SBR sites. Using this R for EcoR1, B for BAM-H1, S for SAL-H1, and a new DNA of 23 kilobases can be inserted here. As I said, using any one this or the combination of RBS or SBR, you can remove and replace 20 kilobase of gene of interest into this lambda phage. Any of these 3 restriction endonucleases can be used to remove the stuffer fragment. Any of these 3 can be used to remove the stuffer region. So DNA fragments with a variety of sticky ends can be cloned. You can use 3, so you can different DNA fragments can be cloned. Recombinant selection with the lambda EMBL4 can be on the basis of size, as we have been discussing, or can utilize a SPI phenotype, SPI. It is in the wild type. These phages are sensitive to PS interference. These are sensitive to PS interference proteins, so that region can be used. Anyway, phage genes responsible for the SPI phenotype are carried on the stuffer region on this type of insertion vectors. Removal of this stuffer region and its replacement by a gene of interest makes this phage recombinant phage insensitive to P2 interference. Means a P2 phage, P2 virus cannot be reinfected here in this region. So these are the features of lambda EMBL4, which is a very best example of lambda phage as a replacement vector. Now, how the cloning experiments with the lambda insertion or replacement vectors are carried out? It follows a certain steps. Now this is the ultimate aim why we are studying bacteriophage as cloning vectors. The cloning experiment with the lambda phage, we can proceed along the same lines as we have done with the plasmid vector. That is how the gene of interest is inserted inside it. The lambda molecules, they are first restricted, new DNA is added, the mixture is ligated, the resulting molecules are used to transfect a competent E. coli molecule. This type of experiment it requires that the vector should be in the circular form. Okay, with the cos sites they should be hydrogen bonded to each other. As you can see, cloning with a circular DNA molecule is done. This is a cos site, the insertion factor is there in a circular form. This eco R1 digested gene of interest is used, and this recombinant molecule used to transfect the E. coli. Okay, although this is satisfactory, this is a very satisfactory process. The procedure is based upon transfection. Usually it is not very much effective. This may be one disadvantage of the whole process, that the transfection is not, means the number of transformants, or the rather than let us say the recombinants which are formed are less in percent. A greater number of recombinants would have been obtained if 1 or 2 refinements are introduced in this gene, and that has been already carried out. You can study it at the higher level of your studies. The what you can say, the refinements which are there, which are used, is to use a linear vector. Simply use, rather than using a circular vector, use a linear vector, and when the linear form of the vector is digested with that specific recombinant restriction endonucleases, the left and right arms are released as separate fragments, as we saw in the earlier slides. Use a linear fragment, take out the stuffer fragments, you have 2 different DNA, and you can use it for end-to-end ligation in a desired manner, in whatever manner you desire. Okay, by this the recombinant molecule can be constructed by mixing together the DNA with the clone and the 2 arms. It becomes easy. As you can see, cloning with a linear DNA as compared to the circular DNA is much more easier. There are various lambda arms, new DNA, various different end-to-end cos sites, they can be ligated, and you can create different chimeras, different DNA molecules. Ligation, it can result into several permutation combinations of the molecular arrangements, and it can include catenanes. Catenane structures are repeated DNA molecules which comprise of the left arm DNA, right arm DNA in repeated sequences. And if the inserted DNA is in the correct size, then the cos site which separates these structures will be at the right distance apart for the vitro packaging. Means it's very advantageous. As you can see here, there is a catenane formation, repeated sites, cos sites, and what you can say, the left and right arm, they are repeated, and your DNA, if it is of the right size, then it can be very perfectly packed in the vitro, and you can have a recombinant lambda phage. Recombinant phages can be therefore produced in test tube, and they can be used to infect a E. coli culture, which is the whole process which we are discussing. And this strategy in particular is very useful to generate large number of recombinant lambda phages, which can give recombinant plaques on your plates. They can be very easily identified by crystal clear plaques or turbid plaques on the plates. So this is how the cloning is carried out using lambda phage as a vector. So in summary, if you see the size of a DNA which can be carried on a vector is, if it is a small plasmid, a standard plasmid, you have the facility of inserting 0 to 10 kilobases of a DNA. I will call it as 1 to 0.1, let us say, to 10 kilobases of a DNA can be inserted in a standard plasmid vector. For a lambda phage bacteria, 0 to 23 kilobases can be used. Now in our next lectures we will be seeing cosmids and some other types of vectors. I will have a small discussion on that in my next video lecture. So this cosmid can accommodate, as I said, as the technology advances further, you have the urge to accommodate larger and larger DNA fragments. So cosmid, it allows 30 to 44 kilobases of a DNA. Bacteriophage P1, which are called as PAC plasmids, this allows 70 to 100 kilobases of a DNA, that is P1 artificial chromosome. Using this bacteriophage, you create an artificial chromosome using human genes also. You extend the gene of interest to 130 to 150 kilobases. That's huge. And if you use BACs, bacterial artificial chromosomes, 300 kilobase maximum can be, gene of interest can be inserted. And with yeast artificial chromosome, 0.2 to 2 MB, million bases. Size increase, megabases, 0.2 to 2 megabases. And you have HACs also, human artificial chromosomes, where the size still increases, and that gives the magnificence of that is the magnificence of the process where we allow larger and larger DNA fragments to create chimeras in the world of agriculture, in the world of microbiology, in the world of pharmaceuticals. We have green biotechnology, we have blue biotechnology, we have red biotechnology. Green for the agriculture, blue for the oceans and the oceanic animals. We have red biotechnology for the human health and the pharmaceutical, all carried out by creating newer and newer genes of interest and creating artificial expression of these genes. Of course, my dear friends, it comes with lot of ethical and legal issues. So we have a different branch of science called as the recombinant DNA studies in legal systems, that is LLB law, etc. Here in these discussions we are interested only in the beauty of technology, in just peeping through the beauty of this recombinant DNA technology, molecular cloning, molecular biology, or genetic engineering. Having said that, my next topic will be discussing some other cloning vectors where in your syllabus only cosmid is there, but I will not restrict my urge to tell you about phagemids, phosmids, YACs, BACs and HACs in my next lecture. So I hope you are enjoying the beauty of recombinant DNA technology. I know it is a bit hard to digest these topics, but if your fundamental knowledge about DNA, genes, genetics and the genomic science is very clear, if you know what is a DNA replication, if you know what is protein synthesis, and if you know the various recombination processes which are natural in the microbial world, I know you are enjoying every bit of this natural technologies which are exploited by the humans in the recombinant DNA technology. So my dear friends, till the time we explore more into this field, till the time I hope you watch and rewatch these videos, you study by seeing the links which I am giving there down below for taking down notes and for a higher level of understanding of the process which is out there going out, and you are having the products like insulin which is derived from recombinant DNA technology, the BT cotton, it may be BT brinjal, it may be golden rice, it may be artificial vaccines, recombinant DNA vaccines which is being produced, and we are observing history today in the time where we have developed a vaccine within 1 year for coronavirus, which is remarkable, and these recombinant DNA technologies have helped us like anything. So enjoy, learn and learn. Be wise. That is what I all wish you for. So till the time we come and meet again for seeing various cloning vectors, goodbye. Have a nice day.